Welcome to the Intelligent Polymer Research Institute. This is the headquarters of the ARC Centre of Excellence for Electromaterial Science. Here at ACES, as we're known, we do fundamental work into the discovery and development of new materials. New materials that can be used in energy and in medical bionics. You'll also see through the course of this tour how we're using the most advanced methods of fabrication in order to develop protocols that will enable structures and devices to be created so that we can take those fundamental advances and use them in those important areas of energy and medical bionics. Our first example will explore the discovery and development of graphene. Graphene, that wondrous material that has amazing properties in the nano world, and yet we've been able to extricate those into the microscopic and the macroscopic worlds in order to realize applications. I'd like to introduce you to Sanjeev.
Hello.
Sanjeev has been working with us in developing and discovering new aspects about the chemistry of graphene. What's particularly important is that we modify the chemistries so that we retain all the amazing properties of graphene, the amazing mechanical, electrical properties, and even biological properties, and yet we make it processable. That is, we can take those fundamental, very thin, small sheets of graphene and make them into structures and into devices. Starting with the discovery of graphite, or the mining of graphite from a natural source, Sanjeev, can you tell us something about the importance of that source of graphite?
Yeah, definitely, Gordon. This is the very high purity vein graphite from Sri Lanka, which is mined, and it is 99% pure. So what we are doing is we are starting with this graphite and making the flakes of this material like this one. And after making the flakes, we are intercalating this material. Between the pi stacking, we put the intercalants. And
so just going back to taking the graphite,
yeah.
So we take the lumps of graphite from a mine.
Yeah.
How do we know how pure that graphite is, or what the properties of the graphite?
We have been characterizing this material by CHNS analyzer, how much is the purity of this material, and also checking the impurities by trace metal analysis, how much is the metal impurity, which are very, very important, how much is the impurity content in this one. Once we ensure the quality, then we start processing the material.
And so we take this lump of graphite,
yeah.
And what's the next step? Is it to exfoliate it into different...
First to make the flakes out of it, and then intercalate those flakes.
So how do we get from here to the flakes?
It is just by manual crushing at this stage. We are controlling the mesh by sieve filtration. And then we do the intercalation of this material.
And that intercalation involves putting other ions between the sheets of graphene in these flakes.
These intercalants need to be of very high boiling nature because they need to stay within the space of the graphite. So we have been optimized, like orthophosphoric acid, nitric acid, and sulfuric acids are the main intercalants which we have been using for integrating the graphite.
So we take this lump of graphite, we grind it into flakes, and then those flakes have multiple sheets of graphene, and you're putting ions in between those sheets of graphene.
Right. Yeah, that's
right.
And then
the next step would be?
Then we give a rapid shock, thermal shock of this to this material so that it pops like popcorn. The volume expansion here, this is 2 grams of material, the volume expansion is between 700 to 900 times. So this much of material expands to this much of volume.
Wow.
So this is such a high expansion of this graphite. Once at this stage, now 7 to 9 sheets of graphene are stacked together. The surface area is very high. Now we can very easily chemically oxidize this material.
Right, so now we've got this thermally expanded material and we're going to chemically oxidize it. And does that cause it to break up into the individual sheets?
Absolutely. This is what we have been getting. This is the graphene oxide, which is a very large sheet of graphene oxide, around 50 to 70 micron sheet, which is having a unique property forming the liquid crystalline nature of the material as well.
So people might think, of course, that 70 micron is not particularly large, but it is. I mean, we're talking about a 70 micron sheet. That's a bit bigger than the diameter of a human hair. But remember, we're talking about a material that's 1 or 2 carbon atoms thick, right?
Yeah.
And so the aspect ratio of that is huge.
Absolutely, very, very huge. Yeah.
And that's what causes it to form those ordered structures in solution, or what we would call a liquid crystalline solution,
Yes,
right?
that's right.
And then what would you do with that once you've got that form of the dispersions?
So we have been using this material for a variety of applications, for spinning the fibers and for coating onto the fabric. This is one example. We have coated this material simply onto this fabric, and this fabric is very conducting. As you can see, it is conducting fabric just by simple coating and reducing the fabric.
So you take our dispersion, which is the graphene, and so this fabric is coated simply by dipping into the graphene solution?
Simply by dipping onto the dilute solution of this material. And after that, it's reduced. Because graphene oxide, there is a lot of defects onto the surface. We restore the aromaticity, and it makes the fabric conducting.
And as Sanjeev mentioned, these same dispersions are what we use for fibre spinning, for creating long lengths of individual fibres, as well as for printing, either in 2 dimensions or 3 dimensions. And we're going to be looking at these processes in later parts of the tour. It's really important that all of those chemistries that we've just been talking about are actually scalable. It's one thing to be able to do chemistry on a bench at a milligram scale or a gram scale, but be able to take those same chemistries and process graphene into tens of grams or hundreds of grams and still retain those special properties has other challenges. So Sanjeev, can you tell us a little bit about the challenges we encounter in scaling those processes for graphene?
Yeah, Gordon, on the laboratory scale, when we are doing the oxidation, we are creating defects, we are putting oxygen, and the material after oxidation is insulator. So how many oxygen functionalities we put in? Now we have to, when we are scaling it up, we have to produce the similar quality what we were getting on the laboratory scale, and that's how we will get the final product which we were using before. So it is very important to optimize the parameters here. So the parameters were easy to operate on a laboratory scale, but it might be difficult to operate it here. So what we have been doing is we are optimizing the conditions of the reactions so that we get the similar quality what we were getting on the laboratory. So there are so many parameters which are involved, so we are optimizing one by one.
So I guess that's why it's very important when we're working on the bench that we keep everything as simple as possible, yet try to maximize the properties of the graphene and the processability of it.
Absolutely, you're right, Gordon. So it is important at that stage as well, the critical parameters, of monitoring those parameters which are critical, and then very careful implementation here to see how effectively we can optimize here as well.
And of course, if we're going to explore different methods of fabrication, the quantities that we make on the bench are never going to be enough, even to explore all the different methods of fabrication we'd like to, right? So scalability is really important.
What we have seen in past as well, Gordon, when from batch to batch variation in the laboratory, but once we process larger quantities, there is minimum batch to batch variation, and we can do very effective research using this material from the same batch. And once we optimize this on this scale, after this scale, we can say the technology is scalable. We can go to industrial scale because at this stage we are doing the reactions at 35 liters. And from 35 liters, taking the batch to around 500 liters or 1000 liters is not that difficult once the parameters are optimized.
So in meeting the needs of our experimental program in terms of exploring fabrication, we're also really making this process ready for industry, ready for manufacturing.
You're absolutely right. So once we are confident on that scale, we can definitely go to industrial scale manufacturing.
Great. Well, we're going to go and have a look at some of the different fabrication methods, the fibre spinning and the printing and the making all of these amazing devices for energy and biomedical applications. So thanks to you guys down here for showing us what we do at a very fundamental level in the chemistry of graphene and how we make it scalable. Thanks.
Thank you.
Hi, Ali.
Hey, Gordon.
So Ali's another of our research fellows who's been working on 3D printing and taking all of the advances that we have in the material synthesis and processability and turning them into real structures. Ali's worked on a number of projects. You've been working on the 3D printed ears, the sleep apnea project, and here we're talking about the stents. So what's important about the properties of a stent for particular, for applications?
One of the important part is about the design first. So we have a couple of different design because we have the ability, to code the G code on the machine, and then send it to this machine. This machine is able to to print the different structure of with a different property, and plus because we have a different material, we can play with the mechanical property. Let's say, for example, if you add the graphene to the polymer, then we can have the better and robust, mechanical property to compare with the normal polymer.
So using 3D printing, because of the flexibility in what we can fabricate, you can actually customize the stent depending on the particular application. And of course, there can be very complicated shapes of stents that are readily realized with 3D printing.
One of the problem, at the moment for the commercial stent is just they cannot, personalize for the patient. By using the 3D printer, we can make it, personalized according to the scan data of the patient.
Right. And we've been talking about graphene downstairs. Of course, it's an amazing material by itself, but it's often blended with other materials in order to improve the properties of that host structure. And I guess that's what we're doing here, right? You're taking small amounts of graphene and blending that through a polymer that would normally be used to create a three-dimensional stent. Is that right?
Yeah, that's true. Actually, it's just it's allow us to use the graphene and make it printable. Like this structure here, and even a small amount of the polymer we can print a stent like this.
And so that graphene gives it extra mechanical properties?
Definitely, yes.
And I guess if it was needed, could also impart electrical properties into the stent.
That's one of the best benefits of using the graphene for the electrode simulation if it's necessary.
Yeah, and all made possible, of course, because of additive fabrication or advances in 3D printing. It's really an exciting time where you can take fundamental discoveries into really practically useful structures almost immediately by you guys working together. Yourself here on the 3D fabrication and Faisal and Sanjeev and you guys on the materials processing.
Yeah, that's true.
Brilliant stuff. Thanks, Al.
No worries.
Hi, Javed.
Hi, how are you?
Oh, very well.
Good.
So we've just been talking to the guys at the early part of our lab tour about the graphene, how we get the graphene from the graphite, and you use all that amazing chemistry, to get those stable dispersions and then scale that process. But of course, you're the one who's weaving all the magic here with those dispersions and turning it into real fibres that we can actually handle. So how does this process work? You get the dispersion. What do you do with it next?
Okay, sure. We starting from here, we make a spinning solution like the honey, and we inject it through the pump. This is like the pump and the solution, and it's go through the tiny spinneret. This is like that. And inject the solution to non-solvent system. This means the solution converted to the solid material.
So by non-solvent system, so we get this solution or dispersion, as it goes into the bath, it basically precipitates, right?
That's true.
It comes together.
Yeah, sure. Yeah. And just in the beginning, this is like the hydrogel shape. And after that, it become dry and become solid state. This is completely, this is like that, or like that.
Right.
You can use this one as a strong fiber, is graphene fiber, based on the wet spun structure.
Right. So it comes out of the bath like that gel state that you just described to us.
Yes.
So that comes out of the bath, then gets collected on the rollers?
This collected through here, and the next step is just improve the mechanical property by the number of the coagulation bath. We can use a stretch it, we can wash it, we can add the other additive you want. For example, any biomaterial you want to coat in the surface of the fiber or improve the electrical or mechanical property, we can do through this step. And we can, of course, we can control the temperature, we can control the tension during the spinning process. It completely optimizing machine. And the end of the process, we can dry the fiber through the heater. There is a heater here,
Right.
and we can collect the fiber. End of the process like that. We can get a spool of the fiber, depend on the material. For example, this is a man-made fiber, polyester we can produce, or graphene fiber through the, depend on the application. We can the single fiber or multi-fiber collected.
So it's really quite a simple, scalable process, but obviously each one of those steps needs to be very carefully controlled to get the properties that you want in the final fiber, right?
Yeah, that's true. Yeah, it looks very simple, all this technology for fabricating material, but it's still powerful, and we can tuning every property you want for future application. If you look at the using for variable technology, of course, you need a superior mechanical property for the fiber. Sure, we have to enhance the property here, and later we can use for the variable technology. If you're looking for the implant the electrode, of course, the other property we can improve here, like the electrical conductivity and biological activity through this process.
And as well as the graphene fibers, I know recently we've been doing work where we're actually spinning long lengths of biomaterial fibers, right?
Yeah.
Where those biomaterials might have a different function from the graphene for particular applications.
Sure, there is in particular application, we use the old one. This is the new material, and we can, of course, convert it to 3D structure through our facility, like knitting, braiding, or other type of the 3D fabrication currently we have.
Hi, Eva.
Hi Gordon, how are you?
No, I'm very well. So we've just been talking downstairs about the advances in materials like graphene and biomaterials, and looking at some of our fabrication aspects. Of course, another important part of our work is the integration of living cells into our printing protocols. Can you tell us something about how we prepare those cells for printing in a facility like this?
So those cells that we have, we culture these cells in this lab. It is a PC2 facility. So we have a risk group rating, which allows us to work in an environment that the cells that as a scientist, we can be protected from those cells, but the cells can actually be handled in a very safe manner. So we have cells which we obtain from human patients, cells that are from immortalized cell lines, cells that are from various tissues that we might be interested in expanding and differentiating down certain lineages. So, so that's where these cells are stored in liquid nitrogen. Here we have a couple of examples of where we store our liquid nitrogen to try and ensure that they're maintained at a very cold, in a very cold environment that allows them to be able to be expanded, taken out of the storage and expanded over a long time. We also have an environment where we try to nurture those cells in terms of providing the right growth conditions to expand. So we have incubators that we if we just go around. So we have here incubators that provides a warm 37-degree environment, a warm 37-degree environment that has air that is good for maintaining a pH. It's an environment that's good for the cells to grow and be conducive to cell growth. So.
Right. And so do you monitor the health of the cells during these processes, or do we do that afterwards?
So quality control of our cells is very important. So knowing that a cell that is derived from a skin biopsy is exactly that in the first instance is very important. If those cells are then reverted to their pluripotent cell state, a stem cell state, we have to identify that those cells do maintain the ability to be pluripotent. So yes, absolutely.
And how do you test that?
So there are a number of markers that we use. proteins that are expressed that are reflective of that pluripotent stem cell state. But we also, more simply, take them out of the incubator when they're grown in the tissue culture plates, put them under the microscope.
Well, let's go and have a look at
put them under the microscope. We have Sarah there, she's looking at some of our cells. Put them under the microscope and ensure they actually have the phenotype that we expect to see.
So under the microscope, is it mostly you're looking at the shape of the cell, or are you actually scanning it to see if there are those markers we just spoke about?
Yeah, so there are different levels of characterization. There's the level of actual just basic morphology, that high nucleus to cytoplasmin ratio that is characteristic of, say, a pluripotent stem cell. But when it comes to looking at those markers, one of the simple methods that we use is immunofluorescent cytochemistry. So we use an antibody that recognises that specific target protein, and if that antibody is linked to a fluorophore, we can see that under the
glows in the dark.
Glows in the dark. We see that under the microscope. We can add a lamp that will mercury lamp that'll excite that fluorophore, and we can see it glowing in the dark. Exactly.
Right. So we get the cells in, we store them, we put them in an environment that they like to stay in, and we characterise them all along the way.
And we grow them. We put them in the incubator to expand, to scale up the number of cells that we need. When we go through the process of bioprinting, we want to create a three-dimensional tissue, not a few cells in there. We want to actually have a high cell to biomaterial mass. And so what we want to do is scale up the number of cells that we have to be able to encapsulate into the biomaterial, which we will do next door. But that process of encapsulation, of working with the cells, it's very important that we do it in a safe environment such as we do in the biological safety cabinets that we have over there.
So are there other characterisation tools for the cells that you use in here, or should we go have a look at the bioplotter?
We can. It's a process of going back and forth. Right. So whilst we scale up the cells here in the first instance, we go to use a bioplotter and we print those cells in a structural form that we'd like to have, but then we bring it back into this lab and we continue the culture and growth. The characterization also extends to, say, a functional analysis of, say, if you're looking at nervous tissue or neural tissue, you'd be looking at multi-electrode array, so look at those electrophysiological responses, or you'd perhaps be using confocal microscopy to look at calcium flux, how those cells network and show coordinated flux.
Okay, well, let's go and see how we build all those cells into the actual 3D printed structure. So having prepared the cells in this sort of environment, we go take them to the bioplotter, and then create that structure. Yeah. But of course, we'd want to bring that structure back into this environment to interrogate the cells again, see if they're still living, if they're proliferating, and maybe even turning into the types of cells that we want them to. Is that correct?
That's correct. Another really very important point is actually demonstrating that the biomaterial that we're actually creating, that nested material, that encapsulated material, is supportive to just basic cell viability. So we have cytocompatibility testing that we also do. That's another aspect.
So if you were to bring the cells here, what would you actually do? You'd load them into this 3D bioplotter?
So
maybe if you just point out how we do that?
Certainly. So what we have is a 3D bioplotter here. There are cartridges that we load our biomaterial that has the cells encapsulated, or just the biomaterial alone. We don't need to have the cells necessarily at this stage encapsulated. We bring that already prepared in a cartridge. That cartridge, almost like a syringe cartridge, that cartridge is loaded onto these cartridge holders on the right-hand side. They are temperature controlled, so it's conducive to bioprinting at particular temperatures that allows the form of the filaments to be preserved.
So the cell, at this stage, the cells would be added to the bioink formulation. Is that correct?
There are two ways we can approach this. We can actually allow cells to be encapsulated into biomaterial, or we can actually seed afterwards. So this is the part where there's an option of one or the other. Having said that, the biomaterial that we bring, that we have prepared under sterile aseptic conditions in the tissue cabinets we have over there, that is then brought here. And yet again, we are in an aseptic environment. So we have a BSc class 2B cabinet that we introduce our cells or our biomaterial with our cells into this environment.
Right. And I know recently we've been successful in printing structures containing induced pluripotent stem cells. That's an amazing advance, isn't it, for both fundamental studies but also potentially for clinical applications?
Absolutely. Pluripotent stem cells have the ability to differentiate down all the different lineages of the body. So we can allow the cells, it's what's quite remarkable is that the cells, which are very difficult to handle in native culture, we can actually incorporate that into the bioink, which allows the viability of the cells to remain viable and to promote the incorporation of the cells into the material. So yes, we can print the material under with the bioplotter into a defined structure, a three-dimensional structure, and then that material, once we take it back into the PC2 lab, we can culture in a media with the growth hormones and other supportive media, additions to that media that allow the cells to differentiate down different lineages.
Right.
So it's quite exciting to be able to create these very high density structures that the cells are able to form a specific 3D tissue.
Yeah, and so that ability to create those structures gives us fundamental insights into how things behave in 3 dimensions.
Yes.
And obviously what we're discovering is it's very different from 2 dimensions.
Yes.
And
We're not a flat body.
Yeah.
We're not a flat surface. Our cells in the native environment in our bodies interact with other cells in that 3-dimensional space, interact with the extracellular matrix, and that biomaterial provides that extracellular matrix that the cells can have support to grow further and to really integrate into the 3-dimensional tissue.
So it must really bring into question the validity of pursuing studies in 2 dimensions in terms of conventional cell culture.
Yeah, it's a growing. It's been a growing concept that 2D is great for fundamental initial studies, but taking these studies, from whether it's the cancer cell biology or basic tissue biology, taking cells from a 2D matrix, a 2D monoculture to a 3D matrix is essential to understand tissue biology.
Great. Well, thanks for giving us that insight into the preparation of the cells for 3D bioprinting and then how they continue to be interrogated after 3D bioprinting in order to understand how they develop in 3D and how we can manipulate their development in 3 dimensions. Great stuff.
Thanks,
Thank you.
Gordon. Thanks for your interest.
Thank you.